Good morning, students.
Good morning.
Good morning. We start our lesson from input control questions. First question, agarose gel electrophoresis. Who wants answer? Answer the question, please. What's your name?
Ержан Еркяем.
Ержан Еркяем. Please answer.
Agar's gel electrophoresis is a method of gel electrophoresis used in biochemistry, molecular biology, genetics, and clinical chemistry to separate a mixed population of macromolecules such as DNA or proteins in a matrix of agars, one of the two main components of agar.
Okay. What kind of devices used for agarose gel electrophoresis? For agarose gel electrophoresis, used horizontal gel electrophoresis chamber. Okay. Next question. Polyacrylamide gel electrophoresis. Who answer?
Можно я?
Okay. Your name?
Amirova.
Repeat, please.
Polyacrylamide gel electrophoresis is a technique widely used in biochemistry, genetics, molecular biology, and biotechnology to separate biological macromolecules, usually proteins or nucleic acids, and the electrophoretic mobility is a function of the length, conformation, and the charge of the molecule. Polyacrylamide gels are based on the theoretical polymerization principle of acrylamide and cross-linking methylene-based acrylates.
Okay. Your name, please.
Amirova Bota.
Bota. Amirova Bota. Okay. Today, the topic of our lesson, laboratory work 12, molecular cloning. Aim of the work, the study the methods of molecular cloning. Plan of the lesson, molecular cloning, and watch the video, DNA cloning. Molecular cloning is a set of experimental methods in molecular biology that are used to assemble recombinant DNA molecules and to direct their replication within host organisms. The use of the word cloning refers to the fact that the method involves the replication of one molecule to produce a population of cells with identical DNA molecules. Molecular cloning generally uses DNA sequences from two different organisms. The species that is the source of the DNA to be cloned, and the species that will serve as the living host for replication of the recombinant DNA. Defining cloning. Cloning is a loaded term that can be used to mean very different things. Cutting off a piece of DNA from one organism and inserting into a vector, where it can be replicated by a host organism, using nuclear DNA from one organism to create a second organism with the same nuclear DNA. For cloning of genomic DNA, the DNA to be cloned is extracted from the organism of interest. The DNA is then purified using simple methods to remove contaminating proteins, extraction with phenols, RNA, ribonuclease, and the smaller molecules, precipitation, and chromatography. Polymerase chain reaction, PCR methods, are often used for amplification of specific DNA or RNA RT-PCR sequence prior to molecular cloning. The fragments then combined with vector DNA and generate recombinant DNA molecule. Cloning a piece of DNA. Plasmid. Plasmid is circular DNA molecule, small circular DNA molecule, and the gene of interest from another organism cuts the plasmid vector with restrictases and cuts the gene of interest from source using the same restrictases and ligate this interest gene into the cut plasmid, and we obtained in recombinant DNA with interest genes. This will generate a population of organisms in which recombinant DNA molecules are replicated along with the host DNA. In molecular cloning experiments, the DNA to be cloned is obtained from an organism of interest, then treated with enzymes in the test tube to generate smaller DNA fragments. DNA genes of interest. Molecular cloning. The vector plasmid with circular DNA molecule, small, under the fragment and DNA genes of interest will be cleaved with restrictases and joined with using ligases and transport into host cells. Recombinant DNA in host cells, and then multiplication and the division of host cell and multiplied bacteria with recombinant plasmid cultivated in nutrient media with antibiotics. In that media, bacteria with recombinant plasmid can be grown, cell division resulting in a clone. In result, we can obtain the clones of these DNA fragments. Recombinant bacteria with DNA, with recombinant DNA obtained clones. Subsequently, the recombinant DNA is then introduced into a host organism, typically laboratory strain of Escherichia coli bacteria. This will generate a population of organisms in which recombinant DNA molecules are replicated along with the host DNA. Thus, both the bacterial population and the recombinant DNA molecule are commonly referred as a clones. A collection of clones that includes all the DNA sequence of a given species is called a genomic library. Genomic library can be screened for clones containing a sequence of interest. The total population of individual clones obtained in a molecular cloning experiments is often termed a DNA library. A genomic library can be screened for clones containing a sequence of interest. Libraries may be highly complex as when cloning complete genomic DNA from an organism, or relatively simple as when moving a previously cloned DNA fragment into a different plasmid, but it is almost always necessary to examine a number of different clones to be sure that the desired DNA construct is obtained. Applications. Molecular cloning provides scientists with essentially unlimited quantity of any individual's DNA segments from any genome. They use it to generate probes that are used for examining how genes are expressed. Obtaining the molecular clone of a gene can be lead to the development of organisms that produce the recombinant protein. Clone genes may be inserted into organisms, generating transgenic organisms, genetically modified organisms. GMO. Gene therapy involves supplying a functional gene to cell lacking that function with the aim of correcting a genetic disorder or acquired diseases. Control questions. Molecular cloning methods, recombinant DNA methods, what is DNA library, gene therapy, and prepare the presentation material, gene therapy, and send me by universe system. Watch the video, DNA cloning. Plasmids. Continue our lab lesson. we see the plasmid and the gene of interest, and added the restriction enzymes for cleave the DNA molecule, genes of interest for cleaved and the cleaved vectors at the same position. And the combined these cleaved parts of DNA molecule, genes, and with cut the DNA molecule, vector molecule, combining with for combining added ligase for join the piece of the gene of interest and with vectors. Ligated DNA is added to bacterial cells. Recombination DNA molecule in the host cell. This process called transformation of DNA molecule in bacteria, then places into petri dishes and the culture. You can see the bacteria containing recombinant DNA and the bacteria with natural vector system, blue and white selection. Trend. Okay. Please check you. 10. Goodbye, students. Our lesson is finished. Goodbye.
Goodbye.